Some of the 2 marks question for this net examination, especially from Gilbert book, and these questions will be very much helpful for the B section. Anagen. It is the hair follicle growth phase. The hair grows in length during anagen. Anencephaly. It is a congenital defect due to failure of the closing anterior neuropore, and because the failure of the closure, the forebrain remains in contact with the amniotic fluid and subsequently degenerates. Angiopoietins. These are the paracrine factors. These mediate the interaction between endothelial cell and pericytes. Anoikis. This term was asked in 18 net examination. I think it was in December. It is a rapid apoptosis which occurs when epithelial cells lose their attachment to the extracellular matrix. Anorectal junction. It is the meeting of endoderm and ectoderm at the anus in vertebrate embryo. Anterior heart field. The heart field cells forming outflow tracts, that is the conus and truncus arteriosus, right ventricle. Anterior interstitial portal AIP. The posterior opening of the developing foregut region of the primitive gut tube. AIP opens into the future midgut region, which is contiguous with the yolk sac at this stage. Important for the alimentary canal development. Anterior necrotic zone. It is the region of programmed cell death on the anterior side of the developing tetrachord limb that helps shape the limb. Anterior visceral endoderm AVE. It is mammalian equivalent to the chick hypoblast and similar to the head portion of the amphibian organizer. It creates an anterior region by secreting nodal antagonists. So again, anterior heart field, anterior intestinal portal, anterior necrotic zone, anterior visceral endoderm, AVE. These are the 4 terms on these slides. Aorta gonad mesonephron region, AGM. This region produces the hemopoietic stem cells. It is mesenchymal area in the lateral plates is planktopleure near the ventral aorta. Next term is the basal transcription factors. These transcription factors especially bind to the CPG rich sites forming a saddle. It can recruit RNA polymerase II and position it for transcription. Bergman glia. These extends a thin processes throughout the developing neuroepithelium of the cerebellum. Brain bow. It is a genetic method to use to trigger the expression of the different combination of amounts of the different fluorescent protein within cells, labeling them with rainbow of the possible colors that can be used to identify each individual cells in a tissue, organ or whole embryo. Brain bow is a process by which the individual neuron in the brain can be distinguished by the neighboring neurons using fluorescent proteins by randomly expressing different ratio of the red, green and the blue derivatives of GFPs in individual neurons. So for detail you can see Gilbert developmental biology book. Causal Ratzier cells. Causal Ratzier cells, CR cells, also known as the horizontal cell or causal, are heterogeneous population of morphologically and molecularly distinct reelin producing cell types in the marginal zone and layer of the developing cerebral cortex in the immature hippocampus of the different species and different time during the embryogenesis and postnatal life. Reelin secreting cell in the neocortex Very important cells these are. Caudal intestinal portal CIP, anterior opening of the developing hindgut region of the primitive gut tube, which opens into the future midgut region, which is contiguous with the yolk sac at this stage. Context dependency. When a role of an individual component of a system such as transcription factor is dependent on the context. What is the context and what will the fate of that organ and fate of that event? It is the context dependency. In the formation of tetrapod limb joints, the same BMPs can induce either the cell death or cell differentiation depending on the stage of the responding cell. CPG islands. The region of DNA rich in CPG sequence is cytosine and guanosine, and in this sequence cytosine and guanosine is connected by a normal phosphate bond. Such islands are formed in promoters as they bind the basal transcription factor that recruits RNA polymerase second. DMRT1. This protein is found in bird, frog and fish. It activates SOX9, which is central male determining gene in vertebrates. DMRT1 is also needed for maintaining testicular structure in mammals. Epialleles, again useful term, variants of the chromatin structure. Variant in chromatin structure that can be inherited between generations. Epialleles are the difference in the DNA methylation patterning that are able to affect the germ cells, thereby transmitting to offspring. So these are the chromatin structure and depends on the DNA methylation. Alpha fetoprotein. In mammals, a protein that binds and inactivate fetal estrogen but not testosterone in both male and female fetus, and shown to rodents to be critical for normal sexual differentiation of the brain. Cyclooxygenase COX2, an enzyme that generates prostaglandins from the fatty acid arachidonic acid. Deep cells, a cell population in zebrafish blastula between the enveloping layers and the yolk syncytial layers. It gives rise to embryo vapor. Deep homology, signal transaction pathway composed of homologous protein arranged in a homologous manner. It is used for same function in both protostomes and deuterostomes. So this slide contains DMRT, epialleles and alpha ferritin. I have discussed. Forward genetics. Genetics, Lunego, limb genes and meltrins. Forward genetics, genetic technique of exposing an organism to an agent that causes random mutation and screening for the particular phenotype. So forward genetics depends on the random mutation, whereas the reverse genetics is depending on the knocking down. The genetic technique knocking down or knocking out or knocking down the expression of gene in an organism. Lanugo is the hair of the human embryo usually shed before the birth. Limb genes. Genes encoding for transcription factor that are structurally related to the protein encoded by Hox genes. Meltrins. Meltrins are metalloproteinases which are involved in the cell fusion events such as fusion of myoblast to form myofiber and of macrophages to form osteoclasts. Reelin, an extracellular matrix protein found in the developing brain. In the cerebellum, it permits neuron to bind to glial cells as neuron migrates and form layers. And in the cerebrum, it directs migration of neuron towards the glial surface. Royalactin, good term, a protein that induces honeybee larvae to become equine. Very important for insect development. Fed to a larva by worker bee, the royalactin proteins binds to EGF receptors in the larva fat body and stimulate the production of juvenile hormone, which elevates the level of yolk protein necessary for egg production. Roundabout protein, roboproteins. These are the receptors for slit protein involved in controlling the crossing of midline of the commissural axon. So this slide contains three terms: reelin, royalactin, and roundabout protein, roboprotein. I think these terms will help you in this examination. I will send you more terms from Gilbert book, and for detail you can search the book. Good luck.